gap between the science that's talked about in really technical terms and the way it applies to people's everyday lives in terms that they can understand. And so this has proved to be really, really important during this coronavirus pandemic. And so one of the first post I did on the coronavirus was this post describing how the test for the coronavirus works. So I'm talking about like the classic RT-PCR tests. So the classic, like sometimes they're called like molecular tests, but these are the tests that look for the viral genetic information. And so this, I did this post, and then it was really awesome because the IUBMB, so the International Union for Biochemistry and Molecular Biology, helped me find translators from around the world to help translate my figures and some of the text into 30 languages that I now have on my blog. And so in the spirit of this like international collaboration in honor of the International Olympics, although here we're all on the same team, I thought I'd do a video explaining how these tests work. I'm going to start with a basic overview of the process, and then I'll go into more details later. So if some of these terms seem unfamiliar, don't worry, we'll go into more details. But the basic idea of this RT-PCR test, so RT-PCR stands for Reverse Transcription Polymerase Chain Reaction, and so we'll get to all of those things. But the basic idea is that the virus has its genetic information in this strand of RNA. When the virus infects a person, it gets the person to make more and more of the virus, which means that the person will make more and more of this viral RNA. And if we can detect that viral RNA, then we can say that the virus is in that person. But this is difficult because even though it might seem like there's like a ton of virus, the amount of viral RNA is still tiny compared to all of our RNA. All of our DNA. So we'll get more into details about RNA and DNA, but they're really similar, and you can interconvert between the two using different enzymes or protein helpers. So basically you can go from RNA to DNA, and DNA from RNA, and DNA to DNA, and RNA to RNA, as long as you use the right enzymes or protein like these helper molecules. And this is helpful because DNA is a lot easier to work with. We have this technique called PCR, Polymerase Chain Reaction, that we can use to make copies of DNA in a test tube. And so this is really helpful because if we can get this RNA out, so with this RNA extraction step, if we can get that RNA out, then we can use this technique called reverse transcription to make a DNA copy of that RNA. And that's more stable, and it's going to allow us to use this PCR technique. So the really cool thing about this PCR technique is that it doesn't just copy everything, which would be super unuseful because we'd just be like copying all of our own DNA, which would be really unuseful, right? Because we want to detect viral RNA. So we need to only amplify something. So amplify, like copy. We need to only copy a region of the DNA that's present in the viral RNA, that's not present in our RNA or our DNA. So it gets kind of confusing because the virus has this RNA, but now we're talking about DNA. So this is a DNA copy of the viral RNA. And so the thing with PCR is you specify what region you want to be amplified. So by amplify, we mean copy. And so basically you use these short pieces of DNA that can stick on to longer pieces of DNA. And then this molecule called DNA polymerase, so it's one of those enzymes I was telling you about, it can copy from DNA to DNA. And so it's going to bind at those ends that you specify, and it's going to copy in between those regions. And so each time that it does this in cycles. So DNA is normally double-stranded, so you have these long double strands of DNA. You do these cycles where you separate the strands, and then those primers, so those short pieces of DNA, they'll bind and they'll bookend the region that you want to be copied. When they do this, so they latch on and then they copy the region in between, then they do it again and again and again. And so each time you're separating those double strands and making new strands, separating in double strands, they're kind of like zippers. So one strand like complements the other strand. So if you know one strand, you can make a copy of the other strand. And so you do this over and over, and each time you're doubling the amount of DNA. So now we're getting a ton of this viral DNA, but only remember if it's present. So if the person's not infected, that viral sequence isn't going to be in the sample that you take, and so you're not going to get anything amplified beyond like a baseline, like noise, just like artifacts, nothing. This baseline threshold, as we'll talk about in a minute. But basically, we need a way to detect that DNA though, because we're making lots of copies of it, but we can't see it, right? It's invisible, at least to our eyes. And so we need to make it visible. And so the way that we can do this is using these probes. So in addition to the primers, we have these probes. And these probes are also short pieces of DNA, but they're special because they're labeled. So on one end, they have the fluorophore, which is a molecule which, if you shine light at one wavelength, it'll give off light at another wavelength. But on the other end of that probe, you have a quencher. So basically, the light that's given off by a fluorophore is a form of energy. This quencher is like, hey, I want that energy, and it steals it before this side can give it off its light. And so these are on opposite ends of this probe. My arms aren't that flexible, sorry. But so you have the fluorophore and the quencher. What happens is, as the polymerase comes through to copy them, it's going to chew up that. So then the fluorophore is getting released from the quencher, and you get light. And so the more probes that get chewed up, the more light you see. And so the probes are getting chewed up, remember, when you're making copies. So the more copies you make, the more probes you get chewed, the more fluorescence you see. And so now that's where we get back to that like baseline thing. So we talk about cycles, remember? So each cycle you bind the strands, so we call that melting. Then you bind the probes, which the primers in the probe, which we call annealing, and then you do the copying, so that's the extension, and then you do it again and again and again and again. So each time you do it as a cycle, each time you double. And so the amount that you will get, the amount of fluorescence corresponds to the amount of copies that you have. The more copies you start with, the faster the fluorescence will rise. And so we define this baseline threshold, which is kind of just like the noise. So that'd be a negative. And we say, okay, if you get the threshold is like the amount of fluorescence that you have to reach to get like over this threshold. We define it as like the number of cycles, and we define it in terms of the number of cycles. So when you're negative, you'll like never get over that threshold, or it'll be like super, super far out. Like it'll take a bazillion cycles before like the artifacts will get high enough to reach the threshold. But if you start with a lot of copies, you're gonna quickly reach that threshold because you have a lot of copies to start with. So you're gonna make a lot of fluorescence really quickly. And this is what we call, this number of cycles it takes to reach that is something called the CT value. So you might have heard of that. So if you have a low CT value, that means it only takes it takes few cycles. So something like usually like 20s, I think, or so cycles to pass that threshold. And then you start like leveling off because you run out of probes and that sort of thing. But so that would be a positive result. If you can still be positive, but have a really high CT count. So you don't have much virus. So you might be like in the very beginning or the very end of the virus. And so some people try to look at like CT values to determine if a person is likely infectious or not anymore. Because remember, this is detecting the viral RNA. It's not detecting live virus. So it's very likely that there is live virus, especially if you have like a low CT count. But what we're detecting is actually just the viral RNA, which is why you can detect the dead RNA or like dead virus on like surfaces and that sort of thing that isn't actually harmful. And so sometimes like this RT-PCR is actually like too sensitive for its own good. So let's get into the details a little more now. Okay, so we talked about, so let's look at what we are starting with. Okay, so this is the SARS coronavirus. So like I said, it's like this membrane-bound sac of viral RNA and a couple viral proteins. And so typically, the RNA that the virus has it has a single long strand of RNA, and it has the genetic instructions, so the recipes for making the different proteins that the virus wants, that the virus needs, such as like this nucleocapsid protein that's going to coat that viral RNA, this membrane protein, that little orange thing that's in the membrane, that's envelope protein that's in the membrane and helps like organize everything and stuff. And most importantly,
well, not most importantly, well, kind of importantly, but we've been talking about a lot is this spike protein, the one that sticks off, and that's actually going to dock onto cells and let the virus get into those cells. And so we can help, we want to detect something, a sequence for something that the virus has, but that we don't have. So basically, the virus has things, the virus is really like simple in terms of it doesn't want to make stuff it doesn't need to make. So it's only going to make stuff that it can't get from our cells. And so we can look for one of those things that the virus makes that we don't make. And so DNA, there's only 4 letters. So DNA and RNA, there's only 4 letters. And so you can kind of have sequences that look pretty similar, or like, because we're using short sequences, remember, like if you have like a random sequences, you'll eventually, like some of them will just happen to match whatever. So we need to make sure that our primers are really specific for this virus, so they're not going to like accidentally bind to something in our own DNA. And so the test makers make a point to make sure that the probes, the primers and the probe that they use are really specific to this virus. And often what they'll do is they'll actually, these tests will do multiple different regions of the virus to be really sure that that you're not getting like a false positive or something. And so like the CDC primers and probes target this N gene, so that's the nucleocapsid protein that surrounds the RNA, but different tests will do different regions. Okay, so now getting back to what we're copying. So remember I said that DNA and RNA are really similar. So they both have the same like phosphodiester backbone, and they have these bases that stick out. So the bases are the unique parts. So there's the 4 letters. So it's A, T, C, and G for DNA, A, U, C, and G for RNA. But the importance of these letters is that they form specific like complementary base pairs. So you have G pairs with C, A pairs with T, A pairs with U, and RNA. And so this is why you can like unzip and zip the strands, and why you can make one copy of the strand from another copy of the strand, because you have this relationship between DNA and RNA where they have these letters that can complement one another. So they both have G and C. And so they differ slightly. In the only place like they differ is in the mostly in like the backbone, like so the sugar. So deoxyribonucleic acid, so DNA, it is missing this OH group that the RNA has. So the RNA is ribonucleic acid, and so it has this ribose sugar, and the DNA has deoxyribose. But the bases, the part that stick off, is the part that's going to actually do this complementary binding, and the bases don't involve that backbone part. So this is why you can have like DNA to RNA and RNA to DNA, because it's involving these base parts. And U and T, so remember the RNA had U, DNA has T, but U and they both can go with A, so that's not an issue that we have to deal with. So the only issue we have to deal with when we want to make a copy of one from another is making sure we use the right enzymes, so the right protein helpers. And so if we want to go from DNA to DNA, you use a DNA-dependent DNA polymerase, and that's what we're doing in the PCR, and this is what our cells do to replicate our genome, so our genetic blueprint. Before, like each time that a cell wants to make another cell, so like copy itself, it has to copy all of its DNA, and so it uses the DNA polymerase. Then for RNA to RNA, so this would be like an RNA-dependent RNA polymerase, and so this is what the virus does to replicate its genome. So it actually has to encode, so it has the genetic instructions for making an RNA dependent RNA polymerase, because we don't have one of those RDRPs. And then, so DNA to RNA, this is what our cells do to make copies of protein instructions. And then RNA to DNA, so this is what we do in reverse transcription. So this is how we make the DNA copy of the RNA. And sorry about the lights. When I stand too still in the lab, the lights go off. 1 second. Okay, I'm back. I don't want to do that. Okay, so we're going to make these copies using this RT-PCR. So reverse transcription PCR and then like real-time PCR. So the RT can mean multiple things, and here we're using both of them, but we already did the original reverse transcription, so now let's talk about the real-time PCR part. So basically what happens is we have those, we make the where did the probe picture go? 1 second, sorry for the technical difficulties. Okay. So the idea with PCR, remember, is that we have those primers, those short pieces of DNA that are specifying the region that we want to be copied. And so you have these cycles of melt, anneal, extend, melt, anneal, extend. So each time you're making another copy. And then I told you about this, this probe. So this is another piece of DNA that binds in the region that you want to be copied. And then the primers will amplify, so they'll copy the region between those two. And when they do, they'll chew up this probe. So it has a quencher at one end and a fluorophore at one end. And when you shine light at the fluorophore at one wavelength, what happens is light is a form of energy. And so you're giving the this fluorophore energy, and then it's going to take that energy and it's going to release it as energy, but it's going to as its own light, but it's going to use up a bit of that energy. So the light it releases is going to be of a different wavelength. So basically the wavelength of light is related to how much energy the light has. And so because it's releasing energy, it's releasing light with less energy because it used some of it, then that light is going to be a different wavelength. And so if you detect that, you set your detector to detect that specific wavelength that you know it's going to emit, then you can detect the fluorescence, so that light coming off. But this quencher's there, and it's like, hey, I want that energy. And so through this thing called FRET, so it's this non-radiative energy transfer, so radiation like light. So this is like a non-light energy transfer. So the quencher kind of steals the energy before the molecule can release it as light, before the fluorophore can release it as light. But it's distance dependent. So this can only happen when the fluorophore and the quencher are close together. When they're far apart, you don't get that. And so when they're far apart, the fluorophore can shine, and you can detect it with your detector on your RT-PCR machine. And so each time that you make a copy, when that DNA polymerase is going to like kind of plow through the quencher and like chew it up, and that's going to separate the fluorophore and the quencher. And so each time you make a copy of the region, you're going to see an increase in light. And this increase in light is going to correspond to the number of copies. And so initially, it's going to take a while, because remember, it's like exponential. So each time you're doubling it. So if you go from, like when you start out, it's like slow going, like you go 1 to 2, 2 to 4, 4 to 8. But then you start like bumping it up, and because you're getting bigger and bigger numbers, so each time you're doubling it faster. And so you get this like exponential curve. So it starts off slow, creeps up, and then and then at the end you like plateau because you run out of stuff. And then so you can't tell it's exponential in the beginning because it's just like hidden in all this noise. So you're always going to get some noise because there's always just going to be like some random thing that binds and makes like a tiny bit of product, where the fluorophore is just going to randomly like break or that sort of thing. But that's not going to like amplify far. And so you're just going to get this little fuzzy thing. But if you have a lot of the viral DNA, or like the DNA copies of the viral RNA, so if you start off with a lot, you're going to quickly reach this exponential phase, because you're like you get a head start. So you haven't seen all those, you start with it naturally high. And so you're already like partway along the curve. So you're going to reach that threshold where you pass the level of the noise really quickly, so in few cycles. So you're going to have a low CT value. So the CT value is the number of cycles you need to reach the threshold, to pass that threshold where it's just noise, and to reach the threshold where it's a true positive. So if you do that, you'll get a positive. If you don't, then you'll have a negative. And so the reason why you have a negative is because the viral sequence isn't there to start with. So remember, you're making copies specifically of the viral sequence. That viral sequence should not be there unless a person is infected or was infected and still has like this like dead viral RNA in them. And so the CT value will be really low if the person is like has a really infectious and active virus type of thing, because there's lots and lots of the viral RNA. And then a high CT count would mean like likely in the early or late phases of the infection. And so that is all I want to say today, but remember that you can find the figures and some of the text in 30 different languages on my blog, thebumblingsbiochemist.com, and I'd like to thank again the translators who've helped me and the IUBMB for helping me find those translation volunteers. And so stay safe, everyone.